Hello friends, today I will discuss the concept of genetic imprinting and uniparental disomy. Genomic imprinting and uniparental disomy are factors that influence how some genetic conditions are inherited. Friend, first I will discuss genomic imprinting. People inherit two copies of their genes, one from their mother and one from their father. Usually, both copies of each gene are active or turned on in a cell. In some cases, however, only one of the two copies is normally turned on. Which copy is active depends on the parent of origin. Some genes are normally active only when they are inherited from a person's father. Others are active only when inherited from a person's mother. This phenomena is known as genomic imprinting. In genes that undergo genomic imprinting, the parent of origin is often marked or stamped on the gene during the formation of egg and sperm cells. This stamping process, called methylation, is a chemical reaction that attach small molecules called methyl groups to certain segment of DNA. These molecules identify which copy of gene was inherited from the mother and which was inherited from the father. The addition and removal of methyl group can be used to control the activity of gene. Only a small percentage of all human genes undergo genomic imprinting. Researchers are not yet certain why some genes are imprinted and others are not. They do know that imprinted genes tend to cluster together in the same region of chromosome. Two major clusters of imprinted genes have been identified in humans, one on the short arm of chromosome 11 at position 11p15 and another on the long arm of chromosome 15 in the region 15q11 to 15q13. Friend, now I will discuss the concept of uniparental disomy, and this concept is very much related to genetic imprinting. Uniparental disomy, in short, sometimes known as UPD, occurs when a person receives two copies of chromosome or part of a chromosome from one parent and no copies from other parent. UPD can occur as a random event during the formation of egg or a sperm cell or may happen in early fetal development. In many cases, UPD likely has no effect on health or development because most genes are not imprinted. It does not matter if a person inherits both copies from one parent instead of one copy from each parent. In some cases, however, it does make a difference whether a gene is inherited from a person's mother or father. A person with UPD may lack any active copies of essential genes that undergo genomic imprinting. This loss of gene function can lead to delayed development, intellectual disability, or other health problems. Several genetic disorders can result from UPD or a disruption of normal genomic imprinting. The most well-known conditions include Prader-Willi syndrome, which is characterized by uncontrolled eating and obesity, and Angelman syndrome, which cause intellectual disability and impaired speech. Both of these disorders can be caused by UPD or other errors in imprinting involving gene on the long arm of chromosome 15. Other conditions such as Beckwith-Wiedemann syndrome, a disorder characterized by accelerated growth and an increased risk of cancerous tumor, are associated with abnormalities of imprinted gene on the short arm of chromosome 11.